Okay, so welcome back to this next video in which we are discussing cyclic AMP signaling. Okay, so we've just been discussing the GS cascade, and we've discussed that a GS coupled G protein coupled receptor can activate GS heterotrimeric G proteins. The alpha S subunit with GTP bound can then go and activate any one of the 9 different plasma membrane adenylyl cyclase enzymes by binding to that C1a, C2a dimer and changing its conformation so that it is now in a conformation where it can actually catalyze the conversion of ATP into cyclic AMP and pyrophosphate, and therefore you're going to start getting cyclic AMP being produced. Okay, then what the cyclic AMP will do is it will activate different targets, okay, but the main target, the most famous and most studied target is protein kinase A. We'll come back to other targets that cyclic AMP can activate later on. Okay, and we've seen that what happens in the activation of protein kinase A is that cyclic AMP binds to those 2 cyclic nucleotide binding domains on the regulatory subunit of protein kinase A. Okay, the cyclic nucleotide binding domain B and then the cyclic nucleotide binding domain A, and that causes the release of the catalytic subunits from the inactive protein kinase A tetramer. Okay, and then they are free to go off and actually catalyze the phosphorylation of serine and threonine residues within proteins. Finally, what we've discussed is that not all serine threonine residues get phosphorylated by the catalytic subunits of protein kinase A. In order to be phosphorylated, a serine or threonine residue needs to occur in this special sequence, okay, which is an arginine or a lysine followed by another arginine or a lysine followed by whatever you like, and then followed by a serine or a threonine. Okay, right. If your serine or threonine occurs in one of these sequences, then it can be phosphorylated by the catalytic subunit of protein kinase A. Okay, now, before then we discuss some examples of where this pathway is extremely important, what I want to discuss is some important concepts. So the first concept I want to discuss is the concept of amplification in this pathway. Okay, now what does this mean? This means that you can go from having only a few GS coupled G protein coupled receptors being activated to having a huge number of different proteins being phosphorylated. Okay, so let's have a look at the steps within this pathway where you get amplification of the signal. Okay, so let's get our picture of the pathway back up again. Okay, so basically, if we imagine having a G protein coupled receptor activated by its ligand, okay, then that G protein coupled receptor can actually activate a huge number of different GS heterotrimeric G proteins, okay, because remember, once it has successfully activated a GS heterotrimeric G protein, both the alpha S and the beta gamma subunit are now cleaved away from it, so effectively it is now free, it's still in this active conformation, and it can now go on and bind another off-state GS heterotrimeric G protein and activate that one. Okay, so it can activate a huge number of GS heterotrimeric G proteins. So that's the first point in this signaling pathway where you get amplification. A single G protein coupled receptor with its ligand molecule bound can cause a lot of alpha S GTP to be produced. Okay, so that's the first point of amplification. Then the next step doesn't contain amplification because it's one alpha S GTP subunit to one adenylyl cyclase. So this can only activate one adenylyl cyclase. Okay, so this can't activate multiple adenylyl cyclases. So there's not an amplification step there, but there is an amplification step now because one adenylyl cyclase enzyme that's been activated can produce absolutely loads of cyclic AMP. Okay, so it can catalyze the conversion of ATP into cyclic AMP and pyrophosphate, but once it's finished, it can do it all over again. So you can produce loads of cyclic AMP. So that's the second site of amplification. The first site was that we got a lot of GS being activated by a single GPCR. Now we're getting that one adenylyl cyclase can produce loads of cyclic AMP. So this is also multiplying up the signal, basically amplifying up the signal. Okay, then there's not an amplification step with the cyclic AMP activating the protein kinase A, because it's 4 cyclic AMP molecules, well actually 2 cyclic AMP molecules to activate each catalytic subunit of protein kinase A. Okay, but then once you've got the catalytic subunit of protein kinase A activated, then there's an amplification step, because this catalytic subunit of protein kinase A, it can go on and phosphorylate absolutely loads of serine and threonine residues. So basically my point here is that from one G protein coupled receptor being activated by one ligand molecule, you can end up with a huge number of proteins actually being phosphorylated, basically, so you can get huge amplification of the signal, basically, and it can cause a big effect within the cell. Okay, so that's that clever concept out of the way. Okay, the amplification of the effect of ligand molecules on the actual cell. Right, so the next thing I want to discuss is how you actually terminate the signal. Okay, so how you actually turn it off. Okay, so let's imagine that our ligand molecule, whatever it is, has now been removed from the extracellular fluid. Okay, so it's no longer activating the G protein coupled receptors. Therefore, the G protein coupled receptors turn off. Okay, that means they are not going to activate any more GS heterotrimeric G proteins. Okay, so the activation of GS heterotrimeric G proteins is stopped now. But how do we turn off all of the alpha S subunits that we already now have in the on state? So that's the first question I want to ask. How do you turn off the alpha S subunit of the heterotrimeric G protein? Well, basically, the alpha S subunit of heterotrimeric G proteins turns itself off, okay, because it is actually an enzyme. It's an enzyme which is capable of hydrolyzing GTP. So if I just draw my alpha S subunit here, okay, so here is my alpha S, and it's got its GTP bound to it, and it's on its own at the moment, or maybe it's bound to the C1a, C2a dimer of our adenylyl cyclase. Okay, right. Now, alpha S subunits of heterotrimeric G proteins, they actually have an intrinsic GTPase. They are an enzyme. They are capable of breaking down GTP to GDP and inorganic phosphate. Okay, so they're capable of hydrolyzing GTP down to GDP and inorganic phosphate. So they are capable of turning themselves off. However, this rate, the rate at which they will actually turn themselves off, the rate at which they will hydrolyze that molecule of GTP that's bound to them and turn it into a molecule of GDP is quite slow, okay? And in fact, what the adenylyl cyclase C1a and C2a dimer is capable of doing to the alpha S GTP subunit when the alpha S GTP binds to it is accelerating the rate at which the alpha S subunit will hydrolyze its GTP down to GDP and inorganic phosphate. So basically, we would say that the adenylyl cyclase enzyme is what's known as a gap for the G-alpha S subunit. So what does GAP stand for? Okay, so GAP stands for GTPase activating, or you can see that A as accelerating, so whichever you want. Activating or accelerating, people will use different words. Okay, the meaning is the same. Protein. Okay, so a GTPase activating or accelerating protein. Okay, and basically what that means is that when the alpha S GTP subunit binds to the C1a, C2a dimer, the rate at which the alpha S hydrolyzes the GTP to GDP and inorganic phosphate is going to be hugely increased. Okay, so that the alpha S subunit is now going to turn itself off much more quickly now that it's bound to the adenylyl cyclase. So let me make this crystal clear. Once the alpha S subunit in the on state is bound to our C1a, C2a dimer here, the rate at which it hydrolyzes its GTP to GDP and inorganic phosphate is accelerated. So it's not just a case of the alpha S subunit activating the C1a, C2a dimer, okay, to start converting ATP into cyclic AMP and pyrophosphate. The C1a, C2a dimer activates the alpha S subunit to start converting GTP into GDP and inorganic phosphate. So it reciprocates, basically. Okay, right. So basically once the alpha S subunit then has bound to the C1a, C2a dimer, it's now going to be activated to hydrolyze that GTP down to GDP and inorganic phosphate. Now once it has hydrolyzed the GTP down to GDP and inorganic phosphate, the inorganic phosphate is released into the cytoplasm and the GDP remains bound to the alpha S subunit. Okay, and now the alpha S subunit is in the off state again. Now when it's in the off state, it can't bind to the C1a, C2a dimer anymore, so it cleaves off the adenylyl cyclase. Okay, so if I draw this, here it is now again. It's now cleaved off of the adenylyl cyclase, and it's going to go back and find the beta-gamma subunit, okay, or rather a beta-gamma subunit. It might not find the identical beta-gamma subunit it was with prior to being activated, but it's going to go off and find a beta-gamma subunit to bind to again. So once it's in the off state and has GDP bound to it, it cannot associate with the adenylyl cyclase anymore, and it wants the beta-gamma complex back. Okay, and when it finds a beta-gamma complex, it will then bind to that beta-gamma complex, and then you're back at the starting point where you have your inactive heterotrimeric G protein. Okay, so that is how we turn the alpha S subunits of heterotrimeric G proteins off. Basically, they are a time bomb waiting to go off in the first place. Okay, but the adenylyl cyclases are capable of speeding up the rate at which the alpha S actually turns itself back off and returns to being in the inactive heterotrimeric G protein. Okay, so to summarize then, when we've removed the ligand from the extracellular fluid, we've blocked further activation of GS heterotrimeric G proteins, eventually our alpha S subunits that are already on are going to turn themselves back off, so that's brilliant, which means that the adenylyl cyclases are going to turn themselves off because they will no longer have the alpha S GTP subunit bound to them, which means we're not going to be producing any more cyclic AMP. But now what we need to look at is how do we get rid of the cyclic AMP we've already created. Okay, so we've turned the adenylyl cyclases off now. That's all been terminated now. Okay, and I should have put a title here. We are looking at signal termination. Okay, but now what we need to look at is how we can actually get rid of all the cyclic AMP that the adenylyl cyclases have already produced. Okay, and this brings us on to the topic of phosphodiesterase enzymes. Okay, so let's discuss phosphodiesterase enzymes. Oops. And there are absolutely loads of phosphodiesterase enzymes, and they're all usually abbreviated down to PDE enzymes. So P for phospho, D for di, and then E for esterase. So phosphodiesterase, and I'll put enzymes. Okay, right. So before I actually tell you the names of all the different phosphodiesterase enzyme genes that we have in humans, okay, I'm going to tell you what they do firstly. Okay, now, not all phosphodiesterase enzymes actually do what I'm about to tell you, which is break down cyclic AMP. Some of them are what are known as cyclic GMP specific phosphodiesterases, and they actually break down only cyclic GMP and not cyclic AMP. Okay, so a very analogous molecule to cyclic AMP, but obviously different in what it does. Okay, and I'll highlight those phosphodiesterase enzymes when we're going through the different phosphodiesterase enzymes. Okay, so the phosphodiesterase reaction with regards to cyclic AMP then involves the breakdown of cyclic AMP. So let me show this. Okay, so I'll draw out my cyclic AMP molecule first. So here's my adenine organic base, represented as that rectangle with an A in. Here's my ribose sugar with the phosphate group between the 5th carbon and the 3rd carbon here to create a cyclic AMP. Okay, and I'll just colour this in. Here is our ribose sugar in green. Here is our adenine organic base in red. Okay, and here is our phosphate group in purple. And basically the phosphodiesterases which break down cyclic AMP are going to convert the cyclic AMP into just AMP. Okay, they're going to break basically this bond that makes the molecule cyclic. They're going to break the bonds between this phosphate group here and the 3rd carbon of the ribose sugar. Okay, and what that's going to create us is just ribose here with a single phosphate group attached to the 5th carbon, and then of course the adenine bound to the 1st carbon. Okay, and this then is just adenosine monophosphate, or AMP. Okay, so let me colour this in. So here is our ribose sugar in green, here's our adenine organic base in red, and here is our phosphate group coming off the 5th carbon. Okay, right, so that's what most phosphodiesterase enzymes are going to do. As I say, a few of them are selective for cyclic GMP over cyclic AMP and will not break down cyclic AMP. Okay, now cyclic GMP is a very similar concept to cyclic AMP. Just replace this adenine organic base here with the organic base guanine, and you've got cyclic GMP. Okay, and of course it can be returned to GMP. Okay, so some phosphodiesterase enzymes only do this one and not this one, but most of them do both, or some of them are just specific for cyclic AMP. Okay, now we're not interested in cyclic GMP, but I'm just going to mention that there because I'm about to talk about how some of these phosphodiesterase enzymes are cyclic GMP specific. Okay, so that's what phosphodiesterase enzymes are going to do. The ones that break down cyclic AMP are going to turn it into AMP. Okay, now, as I said earlier, there are absolutely loads of phosphodiesterase enzymes in humans. Okay, there are 23 known genes, and this continues to rise. Okay, so at present, as far as I'm aware, there are 23 genes for phosphodiesterase enzymes. Okay, and they are grouped into 11 great families. Okay, so I am now going to tell you the names of the 23 genes for phosphodiesterase enzymes and how they are grouped into the 11 families. Okay, so let me start off with the 11 families then. So the 11 families are very simply named. They're named the phosphodiesterase 1 family, or PDE1 family. Then there's the phosphodiesterase 2 family, the phosphodiesterase 3 family, the phosphodiesterase 4 family, the phosphodiesterase 5 family, the phosphodiesterase 6 family, phosphodiesterase 7, 8, 9, 10, and 11. Okay, right. Now, in each one of these families, you have multiple genes. Well, not all of the families have multiple genes in. Some of them only have one gene, but quite a few of the families have multiple genes in. So let me tell you about these different families members. Okay, so phosphodiesterase 1 family then. It has 3 genes within it known as phosphodiesterase 1A, phosphodiesterase 1B, and phosphodiesterase 1C. So to give you an example of how this would be written out, you would write PDE for phosphodiesterase, then you put the family, so you put 1, okay, and then you put the actual family member, so you could put A there, and that means the specific gene basically. That means phosphodiesterase family number 1, gene A basically. Okay, and that refers then to a specific gene product. Okay, right. So let's now go on and have a look at some of these other families and their members. So phosphodiesterase family number 2, that only has one member, and that's phosphodiesterase 2A. Phosphodiesterase family number 3 has 2 members, that's called phosphodiesterase 3A and phosphodiesterase 3B. Phosphodiesterase 4 family has 4 members, phosphodiesterase 4A, phosphodiesterase 4B, and phosphodiesterase 4C and D. Okay, right. Phosphodiesterase 5 family then has 1 member called phosphodiesterase 5A. Phosphodiesterase 6 family has 5 members called phosphodiesterase 6A, 6B, 6C, 6D, and 6H. Okay. Phosphodiesterase family 7 has 2 members, phosphodiesterase 7A and phosphodiesterase 7B. Phosphodiesterase family number 8 has 2 members, phosphodiesterase 8A, phosphodiesterase 8B. Phosphodiesterase family number 9 has 1 member, A, and it's the same for 10 and 11, just 1 member, phosphodiesterase 10A and phosphodiesterase 11A. Okay, and I believe that takes us up to 23 genes now. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. Bravo. Right. Okay, so let me now tell you which of these are selective for cyclic AMP and cyclic GMP and which will break down both. Okay, so phosphodiesterases in family number 1, 2, 3, and 10 and 11, these do both. They break down both cyclic AMP to AMP and cyclic GMP to GMP. So all of the members of family number 1, again I'll highlight these up, all of the members of family number 1, all the members of family number 2, all the members of family number 3, and all the members of family 10 and 11, they all break down cyclic AMP and cyclic GMP to AMP and GMP respectively. Okay, next let me do the ones which are selective for cyclic AMP. So the cyclic AMP selective ones are phosphodiesterase 4s, phosphodiesterase 7s, and phosphodiesterase 8s. Okay, so these are only cyclic AMP. Okay, so I'll underline these ones in yellow. So family number 4, all of its members break down only cyclic AMP. Family number 8 and 7, all of their members only break down cyclic AMP. And then the remaining families, phosphodiesterase 5s, 6, and 9, they are selective for cyclic GMP. Okay, and we'll underline all of those in green. So phosphodiesterase 5, that one member, these 5 members of phosphodiesterase 6 family, and this one member of phosphodiesterase 9 family, they are cyclic GMP selective. Okay, so obviously we're only interested in the ones which either break down both or are selective for cyclic AMP, since we are discussing cyclic AMP signaling, not cyclic GMP signaling. Okay, now the main phosphodiesterases then that you actually find within most cells are phosphodiesterase 3 enzymes and phosphodiesterase 4 enzymes. So these two families are the ones to particularly be aware of, because these are the main ones that you find in cells. Phosphodiesterase 3 and phosphodiesterase 4. The other ones are a little bit more niche. They're found in certain specialized cells rather than being a generic enzyme found in all cells. Okay, right, so these enzymes then, they break down cyclic AMP to AMP and therefore terminate the cyclic AMP signal. Okay, so we've so far seen how to terminate the activation of the adenylyl cyclases. We can turn the adenylyl cyclases off once the ligand has been removed from the extracellular fluid. These enzymes then now are the things which actually break down all of the cyclic AMP that has been produced whilst the ligand was present. Okay, so they now turn the cyclic AMP signal off. Okay, and that means that we are now going to be inactivating our protein kinase A enzymes, because now our protein kinase A enzymes are going to lose the cyclic AMP molecules from their cyclic nucleotide binding domains. Okay, so cyclic AMP is going to come out here. Okay, the regulatory subunits will return back to being in this inactive state. Okay, and I should make sure that you understand that when you activate protein kinase A, okay, when the regulatory subunits bind the cyclic AMP and release the catalytic subunits, the regulatory subunits remain dimerized together. There's no effect on the dimerization here. So the regulatory subunit dimer remains intact even once you've activated protein kinase A. Okay, now once the cyclic AMP is gone, the regulatory subunit dimer returns back into this old conformation where it can now bind catalytic subunits again, and that's going to basically mop up catalytic subunits from the cytoplasm and re-sequester them and re-inactivate them. Okay, so the catalytic subunits of protein kinase A now are going to be turned off because of the destruction of the cyclic AMP signal. Okay, right, so now protein kinase A has been turned off, fantastic, but have we reversed the signal fully yet? No, because we have not yet taken all of the phosphate groups that the protein kinase A catalytic subunits have put onto proteins off. If we truly want to terminate the signal, we need to remove the phosphate groups that the protein kinase A enzymes have put onto their targets. Okay, now, I'm not going to go through all of the different enzymes that are capable of doing this, okay? What I'm going to do is just sort of abbreviate them down and say that they all do this, basically. I'm just going to give you the name of the enzymes which do this, basically. Okay, so, the enzymes then that reverse protein kinase A phosphorylation are what are known as protein serine threonine phosphatases. Okay, and they are the exact opposite of the protein serine threonine kinase enzymes, of which protein kinase A is an example. Okay, they basically cleave phosphate groups off serine and threonine residues on proteins. Okay, and for short, protein serine threonine phosphatases are often abbreviated down to PSTPs for short. So P for protein, ST for serine and threonine, and then P for phosphatases. Okay, so these will now remove the phosphate groups from the targets of protein kinase A, and therefore they will reverse the signal that protein kinase A has been producing. Okay, so now that the protein kinase A is inactive, it's not going to be putting the phosphate groups back on anymore. Okay, so the protein serine threonine phosphatases will successfully now take them off, and they'll stay off, and then the target will be inactivated. Okay, and therefore the signal will truly be terminated once the ligand has gone. Okay, so that's how you reverse this signaling process once the ligand has been removed. Okay, so I'll call it there for this video, and in the next video what we will now do is have a look at some really physiologically important examples of where this pathway is important, okay, which is its importance in hepatocytes and in skeletal muscle cells and in adipocytes in responding to adrenaline within the blood that's been released by the adrenal glands.